Good morning, everyone. I'm Christina from University of Arizona, second-year proficient student. I used to work as pediatric cardiac ICU nurse for 6 years, and very lucky to be interpreter for American professors who give us lectures in the hospital. So let's start my topic today. No closure. My project is supported by NIH. The cardiopulmonary bypass is associated with overproduction of reactive oxygen species. Cardiac myocytes exposed to ischemic react by producing pro-inflammatory cytokines and subsequent activation of leukocytes adhesion cascade, allowing neutrophils to accumulate in the myocardium, adhere to myocytes, and release reactive oxygen species. Despite significant refinement have been made over the years, but oxidative stress and inflammation remain the major concern when using CPB. Under normal circumstances, cells are able to balance the production of oxidants and antioxidants, resulting in redox equilibrium. Oxidative stress can occur when cells are subjected to excess levels of reactive oxygen species or as a result of depletion in antioxidant defense. In this literature, they compare the reactive oxygen species levels in two groups, heart surgery with CPB and without CPB. Because reactive oxygen species seem to be reactive and a short half-life, so it is difficult to evaluate and measure. Instead, they measure the reactive oxygen metabolites, which is indirect marker of oxidative stress. So the first graph showed us derivative reactive oxygen metabolites increase correlates with extracorporeal circulation time in CPB group. In this graph, we can see derivative reactive oxygen metabolites increase correlates with aortic cross-climbing time in CPB group, whereas in group without CPB, the derivative reactive oxygen metabolites does not.
Good afternoon, everyone. It is an
does not correlate with surgical time. The last graph showed us derivative reactive oxygen levels are higher and persistent longer in patients in CPB group compared with the group without CPB. So from previous slides, we can see oxidative stress can occur during cardiopulmonary bypass because of overproduction of reactive oxygen species. And we all know oxidative stress can cause adverse effects on lipids, on DNA, and even cause cell death. So addressing oxidative stress during cardiopulmonary bypass is very, very important for us to provide better myocardial protection. So how? How to address this problem? Obviously, more antioxidants. So NF2, my lab is focusing on NF2. NF2 is antioxidant. It is activated in response to accumulation of reactive oxygen species to bring the balance back to the equilibrium. So for me, for perfusion program, I may think if cardioplegic solution, which is not routinely used in cardiac surgery, can induce NF2 to provide better protection for heart. So here comes my project. My project is to test if NF2 can be induced with normally used cardioplegic solution. Now, what is NF2? NF2 is nuclear factor erythroid 2 related factor. It is transcription factor. It is a basic leucine lipoprotein that regulates expression of antioxidant proteins to protect against oxidative stress triggered by injury and inflammation. Actually, activation of NF2 can result induction of many cytoprotective proteins. We can say downstream target genes. These genes can control process like detoxification process, transcription, signaling, metabolism, and tissue remodeling. But today, I'm focusing on antioxidant defense. How NF2 regulates target genes? It works in many ways, but NF2 keep1 ARE is one of essential signaling pathway. So under normal or unstressed conditions, NF2 strongly binds to its cytoplasmic inhibitor KEAP1 protein and degraded very quickly. So under these conditions, the level of NF2 in nucleus is very low, but oxidative stress can oxidize the residues of KEAP1 residues, leading to conformation of KEAP1 and then releasing NF2. So NF2 translocates into nucleus. In the nucleus, NF2 dimerizes with MAF protein, which is a musculoponeurotic fibrosarcoma protein, and then binds to ARE, its specific DNA sequence, which is antioxidant response elements, and then activate the downstream antioxidant genes, thereby initiating a cytoprotective response. So for my project, I include Donado, high K, low K, HTK, and Seltzer. These solutions were added into SA16 cells, which is adult human myocytes. These are pictures of cardioplegia in my project. There's a Donado, there's high K, low K, Seltzer, HTK. There's the components of each solution. I highlight the final concentration of potassium of each solution, and we can see Donado has highest concentration of potassium, and Seltzer, low K, has lowest concentration. Okay, this is the result I got from Donado. So I treated AC16 cells with Donado at a ratio at 1:4 for 0, 10, 30, 60, 120, and 240 minutes. And then I ran Western blot. Western blot is a method to detect specific protein molecules among a mixture of protein. And I got NRF2 and GAPDH protein expression from cells with Donado. GAPDH was used as internal control. The blue bar stands for the control group. The orange bar stands for the cardioplegia group. We can see the orange bar is higher than the blue bar, so which means NF2 can be induced by Donado. This is the result from high K and low K. I treated AC16 cells with high K and low K, a ratio of 4:1 for different times, and got this result. We can see high K can induce NF2 a little bit, but low K does not. This is Seltzer and HTK. I treated cells with pure crystalloid. Seltzer and HTK just simulation the real condition for clinical patients. Interestingly, we can see Seltzer and HTK seems to suppress the expression of NF2. So we may think one of the reasons is because pure crystalloid treated cells, so cells has no nutrition. So it's kind of distressed, okay? So after getting these results, I'm start thinking why there's so big difference in terms of activation NF2 with different cardioplegia. So the first idea came up is component different. So I tried potassium dose response experiment and got this result. The potassium concentration in this experiment started from 0 to 40 millimole. We can see the final potassium concentration of 5 different cardioplegia in my project are within this range. The highest Donado, and then Seltzer, and then HTK, high K, HTK, and low K. We can see NF2 can be induced very well and constantly with increasing dose of potassium. So my conclusion is Donado and high K have the potential to induce the activation of NF2, and potassium seems to play a central role. The high potassium cardioplegia works very well for decades, but we don't know a lot about the underlying mechanism. So I think maybe this is one of the reasons why patients have good outcome with high potassium cardioplegia for open heart surgery. Okay, that's the end for my presentation. That's the co-authors for my project, and a very special thank to my committee members and co-workers in lab. And Any question?
Jim Beavers, Houston, Texas. Very elegant study, very nicely done. My question is, how did you collect your samples? Was it from general blood, or did you take the samples from the coronary sinus right after the blood exited the myocardium?
It is a cell line experiment. We didn't take blood samples from patients. We just treat our cardioplegia into cells, the cell culture.
So if you took it forward to go to animal studies or to go to human population, how would you collect samples just to isolate myocardium, since the expression of this can be from inflammation and other things? So how can you identify as just the myocardium, would be my question.
For animal experiment, that's the next step for my lab. We'll discuss it with PI and instructors.
Well, thank you very much.
Al Stammer, Sweet Valley, Pennsylvania. A very nice job, and congratulations to your team on continuing to work at this level of research. One of the questions I have with the protein expression, NRF2 protein expression, is related to the mechanism of injury that we know is causing ROS upregulation and release, and that's reperfusion, reoxygenation. Obviously, the model that you have was a continuous longitudinal up until the time period of your, I think it was 240 minutes, I believe was the longest, if I remember the one graph. Did you think about going ahead and, say, entering a burst of oxygen into your Petri plates so that you could see if that would, the NR2 would be suppressed or upregulated?
Yeah, that's what I want to do initially, but because we don't have hypoxia chamber now in the lab, but we'll do it very soon.
Yeah, because you could just burst in oxygen on that and put a higher FiO or an FiO2 of 100% or something.
Yes.
And, okay, well, thank you.
Thank you so much.
We have some questions from the web. From Laura, what was your millimole on the high potassium cardioplegia?
Which one?
The high potassium?
Yeah, high potassium. I think it's a 19.6.
Okay.
The high potassium. Yeah.
Okay, and then a couple more here. Daniel wants to know, was microdialysis used for sample collection?
I think so.
Okay.
Yeah.
And then, let's see. Sonia, were all of these based on a single exposure to cardioplegia solution?
Can I say it again?
Were all of these based on a single exposure to cardioplegia solution?
Yes.
The samples? Okay.
Yes.
Okay. And then Gabriel has kind of a overarching question here. Does anyone have magnesium problems when using custodial?
For my project?
You can just answer for your.
Actually, Custodial, in Banner Health, we don't use it. But I did my rotation in Phoenix Children's Hospital. All patients, pediatric patients, use Custodial, and we didn't find any problems when I did my rotation there.
So Minoli from Boston, and you nicely showed that the expression of NRF2 is increased. I'm sure that increase of amount of protein will increase the activity, but have you actually looked at the activity of the NRF2?
Can you say it again? Sorry.
So you showed that protein amount is increased, but
yes.
you actually didn't show the activity of that protein is increased. So did you do any transcription assay?
Yeah, the next step for me is to explore the mechanism of NRF2, how and why we are working on it now.
Okay.
Yeah.
And I've got one more for you. Sonia wanted to know, let's see. Audrey, was this based on a single dose del nido and repeated high and low cardioplegia? The single dose del nido and repeated high and low?
For the one experiment, we didn't do that.
Okay.
Yeah.
And that was, Sonia, similar question. Were all of these based on a single exposure? to
This
cardioplegia?
single exposure.
Okay. Great.
Thank you.
Thank you.